Let's move on here to begin cellular respiration, and let's start with the first objective, understanding how our bodies use biomolecules to harness chemical energy. And one of the fascinating things about this is we just talked about biomolecules. So this is a really nice continuation. Okay, we eat these biomolecules, we ingest them, right? We eat pasta, carbohydrates, different types of proteins, we eat fats. How do we actually get the energy out of those molecules? That's what we're going to describe here today. And that process is called cellular respiration. It is a four-stage process that starts with glycolysis. And so we're going to look at each of those stages. But we first want to understand, what is the end goal? What are we trying to do overall before we get into the details of the processes? Now, we need to think about ATP first. ATP also is one of the biomolecules we described earlier. This is an example of a nucleotide. And we said when we described nucleotides that they are energy transferring molecules. Their whole job is to take energy, store it temporarily, and liberate it at a different time. So ATP is a medium of energy exchange. And if we look at the structure of ATP, we have a double-ringed. This is going to be a purine here, has two rings. So this is an example of our backbone structure, right? A double purine ring. We have a ribose, which we describe as part of the makeup of nucleotides. And then attached to that, we have three different phosphates, right? Triphosphate. So adenosine, our purine base, the ribose, which is the sugar, right? Our carbon ring sugar here. And then we have three phosphates. So the portion of this molecule that we want to focus on is the phosphate component, okay, the ATP. So the triphosphate means that energy is stored in this part of the molecule, specifically in this third phosphate. When we go from ADP, diphosphate, to ATP, we put some energy in there by adding that phosphate on. Really, really important concept. So the whole purpose of ATP is to move that energy around, specifically in that third phosphate. Let's look at how we actually make this ATP. We have ADP, which again is diphosphate, two phosphates, and we add that phosphate. In adding that phosphate, we also add some energy in there. And so ATP is going to be considered an example of a condensation reaction because we generate water by making that bond, putting that third phosphate onto ADP, putting energy in there with it. And so this is endergonic because we're storing the energy into this molecule, ATP, and the amount of energy stored is about 7 kilocalories per mole. So this is the energy change or the energy stored in ATP by way of this endergonic, also a condensation reaction. There are two mechanisms for us to do that. One is considered substrate level phosphorylation. So we're simply adding a phosphate to ADP, and so there are two ways that we can get that phosphate. Substrate level implies that we're taking a phosphate from another molecule. In this example, the phosphate is on molecule X. We're sort of stealing it off, if you can think about it that way. We're taking it from another source. We're adding it to the ADP, and now we have the molecule without its phosphate and ATP. Okay. We'll talk about some of the different features of substrate level phosphorylation, but it generates a smaller amount of ATP. So it's not as efficient when we talk about the amount of energy that we need to sustain our body. It's not a large amount of ATP. It's also not sustainable, right? It can go for a very short time, and then we need some other major sources like oxidative phosphorylation to be more sustainable. So the second mechanism, oxidative phosphorylation, this requires the electron transport chain. We'll talk all about that coming up. This happens in the mitochondria. This is where we generate phosphate by way of taking the energy of moving electrons down their chain. This is why we have the electron transport chain. So the moving of those electrons down a chain generates the energy to put this phosphate onto ADP. So we're not necessarily taking the phosphate from another molecule. Okay. And oxidative phosphorylation generates much more ATP, much more sustainable, much more efficient. This generates the bulk of the ATP, We'll talk about the numbers in specifics, but of the 38 ATP that we yield from a single molecule of glucose, 34 of those come from oxidative phosphorylation, and only 4 of those come from substrate level phosphorylation. So a much more significant impact in terms of the amount of ATP that we need to sustain our human physiology. Let's look at how we break that ATP down. Again, as I emphasized earlier, we only make ATP to break it. It's only a temporary storage mechanism. It's kind of like the body's energy currency. And the analogy that we gave of our backup charger, we charge it to have it storing that energy so that we can use it later on. So it's only made to be broken down. So the way that we break down ATP is we, through a hydrolysis reaction, we insert water, as we've seen earlier, and then we cleave that third phosphate off and we liberate the energy that's sort of captured in that third phosphate. On the other end of our equation, we have ADP, we have our phosphate that's been cleaved, and then we have the energy that was stored is now released. So this is an exergonic reaction, also a hydrolysis reaction. Okay. So let's look at how we break down glucose. We're going to talk about glucose oxidation as the main mechanism of cellular respiration. I do want to give sort of an explanation here before we get into this. We can break down all of our biomolecules. We can break down glucose, which is the sort of the monomers that are forming carbohydrates. So we break carbohydrates down to their smallest components, glucose, and then we can break that glucose down to liberate energy. We can do the same for lipids. Lipids are broken down to triglycerides, and we can cleave those triglycerides to generate energy as well. Similarly, proteins, we break them to amino acids, and we can use the amino acids to make energy. However, the most abundant source of ATP production is glucose, which means that carbs, right, carbohydrates, are the most important source of our energy and ATP production. So we're going to focus our discussion around glucose, but I want you to understand that other biomolecules and other monomers of those biomolecules can be broken down during cellular respiration as well. But for our purposes, glucose is the most important, the most abundant, the most efficient, really. And so we're going to focus on that as we describe cellular respiration. Now, the overall equation for the energy metabolism of glucose is shown here. So here's our glucose, right? C6H12O6, which is the format for glucose. Here's oxygen, 6O2, 6 of those. And if we put 1 molecule of glucose, 6 molecules of oxygen, our metabolism reaction will generate 6 carbon dioxide as a byproduct, 6 H2O as a byproduct, and then energy. Specifically, 686 kilocalories per mole as a representation of the amount of that energy, right? So we can break glucose down to generate this energy, and this is the energy that we're talking about when we look at that exergonic reaction. This is the energy that we're going to use to store in ATP so that we can have it to generate the energy that we need in our body. Now, before we move on from here, I want to kind of think about this reaction the opposite way. So if we think about inserting energy, inserting water, inserting carbon dioxide, and making glucose, what example in terms of different, where do we know in nature that can actually catalyze this reaction in the backwards direction? What example can we give here? So I'm going to say that in a different way again here. Yes, excellent. Thank you. So plants and photosynthesis. Perfect. Thank you. So you are absolutely correct. Okay, I'm seeing a few responses for that. So plants and photosynthesis is how we generate this reaction in the reverse direction. So plants can take the sun's energy, they can take water and carbon dioxide that we breathe out, and they can use that to make glucose, right? This is how we have glucose stored in the leaves of plants, all parts of the plant, and then it's also going to generate the energy that we need as well. So this is an example here of how humans, not just humans, but most species, can be in a symbiotic relationship with plants. Plants are going to generate what we need, glucose and oxygen. We are going to generate what they need, carbon dioxide. And then obviously the environment can give water and energy. And so they can produce what we need and we can produce what they need. And so this is an example of a symbiotic relationship. Let's actually think about illustrating this, this reaction. So here is our glucose and oxygen. Here is how we liberate, again, carbon dioxide, water from that, and energy. So the energy that we liberate is taken to make ATP. So that energy is captured by adding that third phosphate to ADP and making ATP. Again, the whole reason that we made this ATP is to turn around and break it right back down. So now we break that ATP down, we liberate that energy, and that energy can be used to do cellular work, which is moving things around in the cell, making things in the cell, transporting molecules, hormones, receptors across the cell membrane. And then just that results in bigger functions like muscle contraction, synthesis of memories, right? All of this is really an example of the types of cellular work that we're doing with the release of this energy. Now, I also want to point out here that this is not a perfectly, this is not a perfect process or a perfectly efficient process. We do release a significant amount of that energy as heat. So we are not perfect beings by any means. And so we're not going to capture all the energy that's in that glucose. We're not going to take that and use all of that exactly to do work. We're going to release most of that as heat, and then some of that we can capture as to be used in our body. Now, going back to our equation here, if we take 1 molecule of glucose, we actually need 38 ADP. So while we looked at it in a more simplistic way of energy being released, we can now begin to quantify that energy. How many ATP are we making per single molecule of glucose? So for 1 molecule of glucose and 6 oxygens, we need to phosphorylate 38 ADP and make 38 ATP on the other end of our equation. Now, if we were to look at that in terms of our body in a realistic way, 38 ATP is actually storing 7 kilocalories per mole in each of those ATP, which results in 266 kilocalories per molecule of glucose. Okay, now this is what happens in our physiology. So this is a realistic representation of how much energy actually exists in 38 ATP through our body and through cellular respiration in our physiology. However, if we look at this in an experimental way, right, in a theoretical way, let's say we were to look at how much energy could possibly be released in a single molecule of glucose, it's actually 686 kilocalories in terms of an experimental a way to look at this. So what that means is that the difference between experimental, which is the perfect ideal scenario, and our physiology, which is what happens in our bodies, there's a difference there, right? We only liberate 266 kilocalories when in actuality, scientifically, we know 686 kilocalories should exist in that 1 molecule of glucose. So this is why we say that this is not a perfect process. 420 kilocalories are just lost, right? It's just lost as heat. And so the efficiency of glucose oxidation in our physiology is only 38.8%. In actuality, we get 266, whereas 686 should be there. And so we are only 38% efficient at using the energy that is stored in that glucose, right? About 61% of that is going to be released as body heat. Okay, so having said that, we're going to go on to begin to describe how we liberate that energy. What are the four stages of cellular respiration? What are the four stages of glucose oxidation? And how do what are the mechanisms that we go through to generate ATP from that one molecule of glucose? Now, the three major stages that we're going to look at are glycolysis, the Krebs cycle. The Krebs cycle is also called several other terms that we'll talk about, the TCA cycle, the citric acid cycle, and the third mechanism is the electron transport chain. So the first two, glycolysis and the Krebs cycle, are going to generate ATP by substrate level phosphorylation. Remember, we made the analogy here of taking a phosphate from another molecule, a smaller amount of ATP being made, a less efficient way or less sustainable way to make ATP. The electron transport chain, on the other end, is much more efficient. It is going to be involving oxidative phosphorylation. So that means oxygen has to be involved here, right? This prefix here, oxidative, tells us that this requires oxygen, whereas substrate level phosphorylation does not require oxygen. It can be done anaerobically, which is why this is also called anaerobic respiration, okay? So these are the two major mechanisms of making ATP, and we'll see that different parts of the cellular respiration process, different stages, will make ATP by different means. Okay. Now let's begin by thinking about glycolysis, the first stage of making ATP or breaking glucose down. So we have one molecule of glucose. It's going to be broken down to give us two pyruvate molecules at the end. And this is really a sequence of 10 different enzymatic reactions. So there are 10 sets of reactions that occur to go from glucose to 2-pyruvate. Now, what do we yield along the way? So we insert 2 NAD+, and we reduce those to 2 NADH, which means that we now add 2 electrons to NAD. And just to kind of go back here for a quick second, we talked about NAD as being one of our nucleotides, and its main role is to move electrons around. So NAD plus in the oxidized form without electrons is reduced into NADH, which is carrying 2 electrons. What else do we yield? We yield 2 ATP. So we insert 2 ADP on one end, we insert 2 phosphates on one end, again by substrate level phosphorylation here, and then we yield 2 ATP on the other end. What else is unique about glycolysis is that no oxygen is used. This is not oxidative, this is anaerobic, so this can be conducted in the absence of oxygen. No CO2 is produced as a result. And then this occurs in the cytosol or the cytoplasm of the cell, right? So this is glycolysis, the first stage of breaking that glucose down, where we go from glucose to 2-pyruvate as sort of a transient substrate in our overall cellular respiration equation. So here is the here are the exact steps of glycolysis. And I want to point out a couple things here. You do not need to memorize these, first of all. When you go to grad school, maybe med school, maybe you will have to memorize this step by step, enzyme by enzyme, substrate and product. For our purposes, what I do want you to understand is the overall big picture of what is happening. We take one glucose and we start breaking it down. We start transferring it into different types of molecules. We have hexokinase that comes and cleaves one part of this off, and now we have glucose 6-phosphate. We have phosphoglucoisomerase. It comes and cleaves another part off, and then we have fructose 6-phosphate, et cetera, et cetera, et cetera, until we get our main end product, which is what we're kind of concerned with, which is 2-pyruvates, okay? The second thing we're concerned with here that you need to know is how energy is invested. We invest 2 ATP. And then in the energy harvesting phase of the reaction, we're going to yield 4 ATP. So at the midpoint of glycolysis, we actually end up having aldolase, which is one of our products, being cleaved into two separate G3P. So G3P glyceraldehyde 3-phosphate is actually produced twice for one fructose 1,6-bisphosphate with the help of the enzyme aldolase. So again, the details aren't important, but just know that at the midpoint of this reaction, we end up having two substrates or two products that are turned into sort of two chains of reactions that are happening parallel. So at the end, we end up with 2-pyruvate, 4 ATP, and we invested 2. So we have made 2 net ATP if we subtract the 2 that we invested initially. All right. This is just another representation of glycolysis, again, just showing the actual structural makeup of these molecules and how that is changing as we go from glucose down to pyruvate.